Every time you eat, your body pulls off something genuinely impressive. A sandwich goes in one end as a jumble of huge molecules your cells can't use. And a few hours later, it's been taken apart, absorbed, and turned into energy running everything you do, including keeping you at a steady body temperature while you sit and watch this. That whole journey is what this module is about. I'm going to take you from the moment food enters, through digestion and absorption, and out the other side into metabolism, and how your body defends its core temperature. Let's get into it. Here's the map for the next 20 minutes. By the end, you should be able to do 6 things. Describe how the gut wall is built and how it's wired. Compare the 3 ways the gut is controlled. Explain how carbs, proteins, and fats get digested and absorbed. Summarize what the liver, pancreas, and gallbladder each contribute. Explain what sets your metabolic rate and energy balance. And describe how your body holds its temperature steady. Don't memorize these yet. I'll come back to each one. Just notice the shape. First the plumbing, then the control system, then the chemistry, then what we do with the energy. First, the architecture. Before we talk about how digestion is controlled, we need to know what we're controlling, the structure of the tract itself, and the surprising amount of nervous tissue built into its walls. Here's the big idea that trips people up at first. The GI tract is a single continuous tube, running from your mouth all the way to your anus, about 9 meters long. And here's the strange part. The inside of that tube is technically outside your body. It's open to the environment at both ends. Nothing you swallow is truly inside you until it crosses the wall and gets absorbed. Food travels through in order: mouth, esophagus, stomach, small intestine, large intestine, and out. Then, off to the side, you've got accessory organs: salivary glands, liver, gallbladder, and pancreas. These don't have food passing through them. Instead, they dump their secretions, their enzymes, their bile, their buffers, into the tube that does the work. Now zoom into the wall of that tube. Almost everything along its length, it's built from the same 4 layers, and I want you to picture them from the inside out. Closest to the lumen, the open space where your food is the mucosa, the lining that actually does absorption and secretion. Just outside that is the submucosa, a layer of connective tissue packed with blood vessels and nerves. Then the muscularis externa, the muscle layers that squeeze and push. This is what creates movement. And finally, the serosa, a tough outer wrapper. Same 4 layers the whole way down, just tuned differently in each region. The stomach's muscle layer is beefier. The small intestine's mucosa is folded and fuzzy to maximize absorption. But the blueprint is constant. And tucked inside those layers is something remarkable: the enteric nervous system. Your gut has its own nervous system, roughly 100 million neurons, which is why people sometimes call it the second brain. It's genuinely capable of running local reflexes on its own, without checking in with your actual brain. There are 2 main networks. The myenteric plexus sits in the muscle layer and controls motility, how hard and how often those muscles contract. The submucosa plexus sits closer in and controls secretion and blood flow. Your central nervous system can still dial the whole thing up or down. That's why stress or the smell of food changes your gut activity. But the day-to-day reflexes are handled locally. So the gut can act on its own, but it doesn't act randomly. Everything is coordinated so that secretion and movement match whatever actually in the tube at the moment. Let's look at how. There are 3 control systems, and the easiest way to keep them straight is to ask 2 questions about each. How far does the signal travel, and how fast? Neural control is fast and targeted. Local reflexes in the gut, plus longer loops through the brain, mostly the vagus nerve. Think seconds. Hormonal control is slower and long range. Cells in the gut wall release hormones into the bloodstream to reach distant organs. Think minutes. And paracrine control is local chemistry. A cell releases a messenger, like histamine, that just diffuses to its immediate neighbors. All three are chasing the same goal: match the response to the meal. If you learn only 3 gut hormones, learn these 3. The logic, not by names. Gastrin comes from the stomach when food arrives, and it ramps the stomach up by stimulating acid secretion. Then, that acidic soup moves into the small intestine. 2 more take over. Secretin is released in the response to the acid, and it calls for bicarbonate from the pancreas to neutralize it. Basically a too acidic down here, buffer it signal. And CCK, cholecytosine kinin, is released when fats and proteins show up. It triggers pancreatic enzymes and squeezes the gallbladder to release bile. See the story? Gastrin turns the stomach on. Secretin and CCK get the small intestine ready and set the pace. Those signals drive 2 kinds of movement, and they do different jobs. Peristalsis is propulsion, a wave of contraction behind the food that pushes it forward down the tube. The same motion that lets you swallow upside down. Segmentation is mixing. The gut pinches in several points and then relaxes, sloshing the contents back and forth without moving them anywhere in particular. Peristalsis moves things along. Segmentation keeps them in contact with the enzymes in the absorptive wall long enough to actually finish the job. You need both. Now the chemical toolkit. Digestion is mostly a chemistry problem, cutting big molecules into small ones. And that takes secretions. Let's go organ by organ. The stomach deliberately creates a brutal environment, and it's worth appreciating why. Parietal cells pump out hydrochloric acid, dropping the pH down around 2. That's genuinely acidic. That acid does 3 things. It kills most microbes you swallow. It unfolds proteins so they're easier to chop up, and activates the stomach's main enzyme. That enzyme is pepsin. Here's a neat detail. The chief cells release it in an inactive form called pepsinogen, because you don't want an active protein digesting enzyme sitting inside a cell made of protein. The acid is what converts pepsinogen into active pepsin, and pepsin then begins breaking proteins down. Acid activated, acid loving. It's built for exactly this environment. If the stomach starts digestion, the pancreas does the heavy lifting. It secretes a complete enzyme set for all 3 macronutrients: amylase for carbohydrates, protease for proteins, and lipase for fats. 1 organ, the whole menu. But there's a problem. The chyme arriving from the stomach is strongly acidic, and those pancreatic enzymes hate acid. So the pancreas also releases bicarbonate, a base, which neutralizes the incoming acid and lifts the pH back up to where the enzymes can work, and, as a bonus, protects the lining of the small intestine from being burned. Enzymes to digest, bicarbonate to set the stage for them. Fat has a special problem. It doesn't mix with the watery contents of your gut, so it clumps into big globs, and lipase can only work on the surface. The solution is bile. Here's the key point that shows up on exams. Bile is not an enzyme. It's a detergent. It emulsifies fat, breaking those big globs into a huge number of tiny droplets, which gives lipase far more surface area to attack. The liver makes bile continuously. The gallbladder stores and concentrates it between meals. And when fat hits the small intestine, remember, CCK from earlier? That's the signal that squeezes the gallbladder and releases bile right when it's needed. We've got the tools. Now let's actually break the 3 macronutrients down and get them across the wall. Because remember, until it's absorbed, it's technically outside you. The pattern is the same 3 times. Take a big molecule, cut it into small units across the gut wall. Carbohydrates get broken down into monosaccharides, single sugars like glucose, by amylase and enzymes on the intestinal surface. Proteins get broken to amino acids and small peptides by pepsin and the pancreatic proteases. Lipids get broken to fatty acids and monoglycerides by lipase, with bile helping the whole time. Now, one difference worth remembering for where they go. Sugars and amino acids are water-friendly, so they're absorbed straight into the blood and routed to the liver. Fat products aren't water-friendly, so they get repackaged and enter the lymph first, bypassing that direct trip to the liver. Same digestion logic, 2 different delivery routes. We keep bumping into the liver, so let's give it its moment. Beyond making bile, the liver is the body's central chemical plant. Almost everything absorbed into the blood from the gut goes to the liver first, and it does 4 big jobs. It handles metabolism, processing those incoming nutrients, and, critically, regulating your blood glucose so it doesn't swing wildly after a meal. It produces bile for fat digestion. It handles detoxification, neutralizing drugs, toxins, and the waste products for metabolism. And it does storage, banking glucose as glycogen, plus vitamins and iron. That's why the liver bridges this module. It's where digestion hands off to metabolism. So we've absorbed the fuel. Last question. What does the body actually do with it, and how does it use some of that energy to stay warm? All those nutrients are ultimately fuel, and the rate you burn it has a name, your metabolic rate, total energy used per unit time. The single biggest chunk of that is your basal metabolic rate, the energy you'd burn just lying still, keeping the lights on. Heartbeats, breathing, brain, cell maintenance. On top of the basal rate, physical activity adds more, and so do smaller factors. The energy it takes to digest your food, your body size, and hormones like thyroid hormone. Then there's energy balance, which is beautifully simple. It's just intake versus expenditure. Take in more than you burn, and the surplus gets stored, and you gain. Burn exactly what you take in, you're stable. Burn more than you take in, and the body draws on its stores, you lose. Everything else is details on top of that one equation. And here's where metabolism connects to temperature. Burning fuel releases heat, and your body uses that to hold a core temperature right around 37 degrees Celsius. The thermostat is your hypothalamus, and it works by negative feedback. It defends a set point. When you get too cold, it acts to conserve and generate heat. You shiver. Your blood vessels constrict to keep warm blood deep in the core, and your metabolic heat production ramps up. When you get too hot, and it does the opposite to shed heat, you sweat. Your vessels dilate to bring blood to the skin, and metabolic heat drops. Cold, generate and hold. Hot, release. A thermostat made of biology. So that's the arc. One continuous tube with 4 layers and its own nervous system. 3 overlapping ways to control it. A secretion toolkit from the stomach, pancreas, liver, and gallbladder. The same break it down and absorb it logic for carbs, proteins, and fats. And finally, the fuel we absorb feeding our metabolism, which, among everything else it does, keeps us at a steady 37 degrees. Go back to those 6 objectives at the start and see how many of those you can explain out loud. That's your best study check. Thanks for watching. Make sure to subscribe for more, and I'll see you next module.